Good afternoon. Welcome again, the present audience and the virtual audience. I would like to introduce you Aurélie Goenval. Dr. Aurélie Goenval is a distinguished expert in the field of genetic therapies for neuromuscular diseases. She's based at the Université Versailles, leading groundbreaking research projects on the use of antisense oligonucleotides technologies, focusing on exon skipping strategies for Duchenne muscular dystrophy. Her first studies of her early career stages were published on Science, and in which Aurélie and her colleagues showed that it was possible to rescue dystrophic muscles using proper RNA-mediated exon skipping, as you will learn about it very soon. Aurélie's expertise lies in developing innovative molecular tools that target specific genetic mutations, providing a foundation for personalized medicine. Dr. Goenval combines deep knowledge of molecular biology with practical experience in preclinical and translational research, which is significantly advancing therapeutic applications for rare genetic disorders, such as the Duchenne muscular dystrophy. She is highly active in projects, project management and collaborations of European and international grants, like the Darter Coast, which has been a very successful European network of scientists working in the area of RNA therapeutics. She largely contributes to the advancement in biotherapy innovative strategies, so please join me to welcome Dr. Aurélie Goenval, who was kind to accept, please, our invitation to come to our lab and talk to us about the exon skipping approach for treatment of Duchenne muscular dystrophy. Thank you so much.
Okay. Thank you very much, Mirella. Can you hear me well if I don't take the mic? Yes? Or do you want? I can hold it?
I think it's possible to do that.
Okay. Let me know. You can check, and if it's not good, I will take the mic. So thank you very much for the introduction and this invitation. It's a great pleasure to be here. It's only my second time in Brazil, and it's very nice to meet all of you. So today I feel a bit like an imposter because I'm not talking about retina or vision. I will really talk about gene therapies and exon skipping therapies. So this is the outline of my presentation. I will give you a little overview of antisense technology and what we can do with antisense sequences. Then I'll go to the principle of exon skipping for Duchenne muscular dystrophy, then talk you through the preclinical development of the tools that we've been using, some strategies that we're currently developing to improve even further this strategy, and finishing by the work that we've done in collaboration with Cyril in the brain. So antisense technology is really based on the use of what we call antisense oligonucleotides. So we can abbreviate them using ASO or AONs, depending on the literature that you will read. They're basically nucleic acid oligomers of between 12 and 30 nucleotides, depending on the chemistry. And they are typically synthetic, so they are modified to be resistant to nucleases, and they modulate RNA expression by binding to mRNA. It can be messenger RNA or even non-coding RNA. So they can modulate the expression depending on different mechanisms. We can use either what we call gapmer antisense oligonucleotides, and that will lead to the degradation of RNA, the targeted RNA. Or they can use a steric blocking mechanism, and in that case you can do translation inhibition, gene activation, inhibition of RNA toxicity, or splicing modulation. So this is illustrated here. The first strategy, which is called gapmer. So antisense oligonucleotides are called gapmers when they are only modified here. You see the blue parts. Here they will have chemical modification that protects them against nuclease degradation. And in the middle, here they will have a DNA gap, which means that it creates an heteroduplex between DNA and RNA that will recruit RNase H and degrade the targeted mRNA. So that's an alternative to siRNA, for example. And so this is a strategy that's highly used, for example, by Ionis Pharmaceuticals that have commercialized several antisense oligonucleotides on the market. And we've also used this technology with the chemistry that we're doing. We've been using in the lab, targeting Huntington, for example. The other mechanism, the steric blocking mechanism that we are mostly interested in that case, the oligonucleotides are what we call fully modified. So the entire oligonucleotide is chemically modified. And in that case, you can use it either for inhibiting translation. You see, these were really old papers because it was one of the first applications where oligonucleotides were used. It was quite simple when you think about it. It was just designing an antisense oligonucleotide that would bind to the AUG and inhibit translation. More recently, an opposite strategy has been developed using antisense oligonucleotide, and that's to induce gene activation. And this is because most genes have a primary AUG, but they also have what we call upstream AUG that are not used. But sometimes the ribosome starts here, which tends to decrease a little bit the main use of the AUG here. So if you block some of the upstream AUG, you will enhance the main use of the primary, and that tends to increase gene expression. So it was first published in 2016, and since then there has been several other publications. Another example I want to mention is the inhibition of RNA-mediated toxicity. That's the mechanism that's used in myotonic as a therapeutic strategy for myotonic dystrophy. As you may know, myotonic dystrophy is caused by the toxicity of the RNA that includes lots of CUG repeats in the 3' UTR of the DMPK transcript. And so it sequesters a lot of proteins here, and the fact that these proteins are sequestered by the RNA makes it toxic because they cannot play their role, especially in splicing. So here, the role of the oligonucleotides will simply be to compete with the hairpin, and so that will tend to free the sequestered protein, and that will inhibit the toxicity, and that serves as a therapeutic mechanism. And the final mechanism that we are mostly working on is splicing modulation, and in that case, you use antisense oligonucleotides to mask splicing signals, and then you can either induce exon skipping or exon inclusion. And this can be used either as a therapeutic purpose to do splice correction. You can use it to interrupt an open reading frame. So if you skip an exon, then you may interrupt the reading frame and induce downregulation of a gene. Or finally, if you want to play with isoforms and play with the with the alternative splicing, you can do isoform switching. So, as I mentioned, exon skipping has been mostly used for Duchenne muscular dystrophy. Exon inclusion is used, for example, for spinal muscular atrophy. You may know there's already an antisense oligonucleotide drug approved, and that was really life-changing for patients with SMA called nusinersen. And that's doing exon inclusion. That's also in use for Pompe disease. And different genetic tools can be used to induce these splice switching interaction. You can use either antisense oligonucleotides or vectorized sequence in AAV vectors, and I will mention both. But I will focus today on exon skipping for Duchenne muscular dystrophy. So we've heard a lot about Duchenne yesterday, so I don't need to spend much time on it. You already know that Duchenne muscular dystrophy is caused by a mutation in the DMD gene. It's the largest human gene. It's encoded by 79 exons. And the neuromuscular and cardiac manifestations of the disease are due to the loss of the muscular isoform, so the DP427M, which is represented here. So this is a full-length, a very long isoform that plays a crucial role in the muscle, and the absence of dystrophin in the muscle and the entire dystrophin-associated complex induces muscle degeneration and the fatal outcome that we know. So the exon skipping concept emerged several years ago already now, and it came from the observation of the mutations that were present in DMD, because most mutation presents deletions of one or several exons, and also that mutation in the DMD gene can cause another myopathy, Becker muscular dystrophy, which is much milder. And this is here an example of a DMD patient that carries a deletion of exon 50. And you can see that exon 49 and 51 are not in frame, which will induce a premature stop codon and a truncated protein that's not functional. So the idea of exon skipping is to exclude an additional exon to restore the reading frame. And in that case, if we exclude exon 51, we can join 49 and 52 and restore this reading frame. That will lead to a protein that's slightly shorter internally, but it's functional and that's very similar to proteins that are found in Becker muscular dystrophy. So really the therapeutic concept is to convert a severe phenotype of Duchenne towards a milder phenotype of Becker. It's not a complete cure. This is important to know. So the exon skipping can be induced using antisense sequences, and the main way and the initial way that was used was to develop antisense oligonucleotides, and I will come back to that part in the second part of my talk. Different chemistries have been used in clinical trials, and they had shown results, but one of the prerequisites is to keep injecting these oligonucleotides. So when I was doing my PhD a long time ago, I was working in Genethon, and this is a gene therapy association and an institution that had a very strong background in AAV gene therapy. There are very good viruses for muscle delivery, so we wanted to take advantage of this. And so we wanted to deliver the antisense sequences using viral vectors. So that's what we did. We had to use a cassette to encode that antisense sequence. So we used the U7 snRNA. So U7 is a small nuclear RNA, similar to the small nuclear RNAs that are involved in splicing, except that the U7 is normally involved in the maturation of histone pre-mRNA. So it looks like this. It has a little loop. And here it normally has an antisense sequence to the pre-mRNA for histone. And it had been shown that you could actually change that sequence here to make it specific to any gene that you would want. And that would be a cassette to shuttle antisense sequences. So we modified that antisense. We cloned it into an AAV vector and produced AAV encoding this. And that was, I will show you now. That had a lot of advantage because it colocalized with pre-mRNA and it induces a real stability of the antisense sequence. As a proof of principle, we worked in the MDX mouse model. So the MDX has a nonsense mutation in exon 23, so that's a stop codon. Luckily, exon 23 is in frame, so if you exclude, so here that's the truncated protein that's induced with this mutation. So if you exclude exon 23, you will join 22 and 24, and you will induce what we call a quasi-dystrophin. So you see here, it will only have a very small deletion here, and in the MDX, we know that this is a very functional protein. So we constructed a U7 here that was specific of this exon 23, so to force its exclusion, and we've produced the vectors. And you see, this is old results, but it was really groundbreaking at the time because we had a very high efficiency of that strategy. This was the beginning of the antisense technology, where people were using antisense oligonucleotides. And when you were using antisense oligonucleotides, you had only maybe a couple of positive fibers, maybe 5–10%. And here it was like 90% of the muscle was really dystrophin positive. So this was really encouraging. And you can see here by Western blot, it was like really the levels of one-type muscles. So we did a lot of studies with the AAV U7. We worked also on a more severe mouse model called the double knockout that also has the eutrophin invalidated. And we also did proof of principle in the dog model, the GRMD dog model, which is interesting because you need to skip 2 exons to restore the reading frame in the dog model. We also demonstrated the feasibility of doing multi-exon skipping in another mouse model, and more recently working with U7 targeting the exon 51. So the system has a lot of advantages. This AAV-U7, it can induce particularly efficient exon skipping and a long-term restoration. So you do only 1 injection and it lasts for several years. It can deliver multiple U7 and induce multi-exon skipping. It's compatible with systemic delivery. And depending on the AAV serotype that you will use, you can target the heart very efficiently. For example, a serotype 9 of AAV vector injected intravenously goes very, very well to the cardiac muscle. There's a clinical evaluation of this strategy in the US at the Nationwide Children's Hospital in Ohio. And that's a phase 1/2 clinical trial that was conducted in 3 boys. And they had an exon 2 duplication. So this mutation has the advantage of when you get rid of 1 exon, you have a full-length dystrophin. So this is a real advantage. And they got really positive results. There are challenges with this strategy, and these are the same challenges that gene therapy face. You are using AAV vectors, so there's an antibody response against the capsid, so you cannot go back. You cannot inject again. So eventually when dystrophin starts to go down, and we've seen that in the dog model, where after 5 years we could see that we were starting to lose AAV genomes and the dystrophin levels were starting to drop down. And then you cannot re-inject the AAV. So that's a problem, and a lot of research groups are now trying to find ways to be able to re-inject. So we started working with antisense oligonucleotides for several years when we were facing these re-injection problems. And oligonucleotides, so the first trials for Duchenne were reported in 2007, and that was using antisense oligonucleotides with the 2' O-methyl chemistry and the PMO. So as you may have heard, lots of programs have terminated because they were not working sufficiently, or they had little side effects. But there are today 4 drugs that have been approved for DMD, well, at least by the FDA and the Japanese agency, not in Europe. And they are all PMOs, morpholino, targeting exon 51, 53, or 45. They only induce a few % of dystrophin restoration, so it was a real controversy. Their approval was a controversy. So clearly there's still room for improvement. So there are lots of challenges that remain. We need to improve the efficacy of the exon skipping, and that mostly goes through improving targeted delivery. We need to have a better delivery to the target tissue, and the target tissue for Duchenne is the entire muscle, the entire skeletal musculature, but also the cardiac muscle and the CNS. So that is a lot of target tissue. There are a lot of new developments in the field. The fact that there's 4 approved drugs didn't discourage the companies to go even further. There are, I think, more than 20 clinical trials and looking at the new generation of antisense oligonucleotides. They're peptide conjugated, antibody conjugated, people working with endosomal escapes, and also alternative chemistry. So there are lots of reviews out there that you can go have a look about these new strategies. What we've worked with is what falls into the category of alternative chemistry. So for several years, we've worked with a chemistry of synthetic DNA, which is called tricyclo-DNA. It was originally created by Christian Lehmann at the University of Bern. And this is conformationally constrained. You see there's 3 additional carbons. And what is interesting to just remember is that it has very specific and interesting properties for therapeutic oligos, because it has an increased affinity towards the RNA. So it's very sticky to the RNA molecule. And it also has an increased hydrophobicity. So several years ago, we've published a study that was comparing in the MDX model the efficacy of the tricyclo with the two other chemistry that were used in clinical trials, so the 2'-ormethyl and the PMO. And you can see here, these are the exon skipping levels in the different muscle and in heart, and here the different levels of dystrophin restoration. And tricyclo were able to really induce much higher levels. And this was associated with restoration of muscle function here, restoration of the respiratory function, also cardiac function. So it had a really nice therapeutic benefit. So we took forward the preclinical development of this chemistry. It was a lot of work for a lot of years. Initially, the tricyclo presented a very safe, so they had never been used in humans or anything. So we had to be very careful with toxicology. They presented a safety, an encouraging safety profile, but as most of the oligos at that time, they were made with phosphothyrate linkages. So instead of having phosphodiester link, they have phosphothyrate. And that normally induced some side effects. So in particular, we were looking at urinary biomarkers. And here you can see that in some of them, we were starting to see an elevation after 3 months of treatment. So really what we wanted to develop at that stage was an antisense, a tricyclo candidate that would not have phosphothyrate linkages. And so we tried, because tricyclo are very stable also in their phosphodiester. So here, basically it's all the linkages. You can either have here an oxygen or a sulfur. And having the sulfur is mostly useful for biodistribution properties because it really binds strongly to albumin and a lot of protein in the serum. And for other chemistry, it's also used for stability. But for tricyclo, we didn't need them for stability. So we started looking at tricyclo that had either 0 phosphothyrate linkages or 100%. And you can see here that it clearly correlates with the tissue biodistribution. So when you have no phosphothyrate, well, it doesn't distribute very well. So, and that's inversely correlate with efficacy. So the one that induce strong exon skipping are the one that really have a lot of phosphothyrate. So really, as I said before, it correlates with the protein binding. So this is showing you the protein binding in the plasma. So it binds, you see that tricyclo that has all its phosphothyrate linkages binds to lots of protein, whereas one that has no protein. So it's mostly peed off, let's say. So clearly, reducing the PS content affects biodistribution and efficacy. So we had to find another strategy because we really still wanted to get rid of this phosphothyrate. So we decided to link the oligo to a lipid, a fatty acid, and in that case, we decided to conjugate it to palmitic acid. And so we compared a lot of compounds, either one that was containing phosphodiester or phosphothyrate with the lipid. And to make a long story short, because I'm not going to take you through all the developments, but we ended up developing this new generation of tricyclo that's now completely free of phosphothyrate linkages, but it's linked to a palmitic acid, and that presents a much higher therapeutic index. So it's very safe. We did some preclinical studies in non-human primates in partnership with Sky Therapeutics, which is now exclusively producing these oligonucleotides. And we had a very favorable tox profile, very efficient exon 51 skipping. So based on these results, there was a clinical trial that started last year in France under the sponsor of Sky Therapeutics. So there was 12 DMD patients included. And so the phase 1 is completed now, and Sky Therapeutics is starting the phase 2 for this trial. So this is very exciting. What we're still doing in the lab is trying to still improve this technology, but not necessarily working on the compound itself. So we've already seen what we can do on the compound, trying to link it to a lipid. but we also can play on cellular mechanism to improve the strategy. And there are common challenges that are common to all oligonucleotides, mostly the fact that they are stuck in the endosomal compartment, so they do not escape the endosomal very well. So we're working on this. And there are also challenges that are very specific to DMD, and I will mention 2 examples that we're doing in my lab. One of them is trying to increase the level of RNA that's available for the exon skipping to occur. And the other one is trying to improve angiogenesis. So the first one is, and this is a challenge that's specific to DMD, it's the limited amount of dystrophin mRNA that's available. It's been shown that in DMD that the level of transcript are decreased compared to wild-type levels, and it was long thought that it was due to nonsense-mediated decay. But more recently, it was shown that it was not necessarily only due to the nonsense-mediated decay, but that there is what we call a 5', 3' transcript imbalance. So if you look at the level of transcript along the gene, so at the beginning of the gene, the level of transcription in white, this is the MDX level, and in black, it's the wild-type level. You have similar transcription initiation, but towards the end, you can see that there's much less transcript available. So when you want to skip an exon that's around here, it means that you have maybe twice less transcript available to do this. So, and it was shown again in previous studies that this was due to epigenetics. So there was histone marks on the chromatin suggesting that the DMD chromatin, when it was mutated, was less prone to transcription, for example, in the MDX. So we made the hypothesis of increasing these levels, maybe using HDAC inhibitors. So we worked together with the Netherlands, with the group of Aartsma Roos and Spitali, trying to use HDAC inhibitors. So we've tried different type of HDAC, givinostat, valproic acid, or another one. And we did co-treatment with antisense oligonucleotides and the HDAC treatment. And here, at the end of the treatment, we were looking at the dystrophin restoration. And you can see that with the different muscles, overall, when we added the HDAC inhibitor treatment, we obtained an increase in dystrophin restoration. And this was due here, for example, with the VPA to, indeed, we did not correct the 5', 3' imbalance. You can see it still decreases. This is another representation of the bar graph. But at least we had more RNA level overall. So more RNA levels means that you will have more copy available to make more protein. So that was the reason why we observed more dystrophin. And here is a similar study that we did on the long term to be able to do functional tests. So we selected valproic acid, and again we did like a 3-month study. And here this was the level of exon skipping. You can see that when it's treated with both oligonucleotides and acid valproic acid, we had an increase, and that was translating into more dystrophin protein. Here again on the immunohistochemistry, and this this translates into a phenotypic improvement, or at least here with the biomarkers. You can see that we normally check the levels of myomycin 3, which is a biomarker of dystrophic animals and dystrophic patients, and we had a much better outcome and nice significant decrease with the double treatment. And here is a functional test on the inverted grid. So these results suggest that combined therapies using exon skipping and HDAC inhibitors can be useful. The final thing that I want to mention about trying to improve this strategy is working on angiogenesis. So it has been shown in patients as well as in mouse model that the dystrophic muscle presents deficits in microvasculature. So there are structural and functional deficits. This can, for example, this is what we've looked at in MDX muscle. You can count the number of blood vessels per fiber, so you can really see how the muscle is vascularized. And there's less, here there's less blood vessel per fiber in dystrophic muscle. So these deficits not only contribute to the histopathology itself, but it also limits the biodistribution of therapeutic compounds. So when we want to inject oligonucleotides, if the muscle is less irrigated, then clearly you would have less compound reaching the muscle. So we made the hypothesis that if we could rescue some level of better angiogenesis, then we would deliver better. And as a proof of concept, we used a gene therapy trying to increase angiogenesis. So in the muscle that were treated with this AAV, you can see that we were able to restore a number of vessels per fiber that was really close to the wild type levels. And in that case, then the biodistribution of the oligonucleotide is much higher in the target tissue, and that induced much higher exon skipping efficacy. So the hypothesis is somehow verified, and now we're trying to develop more translational methods to improve angiogenesis than necessarily gene therapy. So now I want to move to the CNS part, and these projects started even before the big comparison study that we did. But one of the specificity of tricyclone that we identified in the comparison study when we compared them with 2'-ormethyl and morpholino was that they were able to cross the blood-brain barrier a little bit. So when we looked at exon skipping levels in hippocampus, cortex, and cerebellum, we were able to detect exon skipping there. And this was the first time that a naked chemistry of oligonucleotides was able to cross the blood-brain barrier. You can see that none of the others were detectable. This was inducing a little bit of restoration in the brain. You can see here on the Western blot, and you probably don't see, but by immunohistochemistry, you can also see dystrophin restoration. And because we were working with Cyril, and Cyril had all these tests available in these behavioral tests in mouse model, and he had already shown that the MDX shows some level of deficits. We did some tests already at that stage, and in particular we did at that stage the unconditioned fear response, so constrain the mice and look at the fear response, because the dystrophic mice, and Cyril will go much more in details, I think. So they show about 80% of freezing response after this stress, whereas the wild type, they only show 10%. So when we did this test with the treated mice with the tricycle, we were able to see a complete restoration of this phenotype. So this was really quite surprising considering the low levels of dystrophin that we had. So of course it was very encouraging, because it suggested that some of these deficits could be reversible eventually in patients, but that also raised a lot of millions of questions that we wanted to ask. We wondered if other neurobehavioral and cognitive deficits could be rescued, and if so, what levels of dystrophin were required. Was there a minimum level? What would be the impact of other dystrophin isoform or other gene products restoration, as Alvaro would say. Alvaro, if you're listening, you see that I corrected myself for you. So we embarked into a large European consortium called BIND for brain involvement in dystrophinopathies. That was led by UCL in London, that had a lot of different European partners. And one of the objectives of that consortium was to better understand the brain comorbidities to eventually guide future treatments. As you know, and it was originally even reported by Duchenne de Boulogne in 1868 that some of the patients displayed cognitive defects, but now it is estimated that nearly 50% of patients have some levels of neurobehavioral or neurocognitive phenotype. They can manifest with very variably, so can go from language disorders, intellectual disability, reading disorders, autism spectrum disorder. They can be classified in 4 main domains: neurocognitive, emotional, academics, and neuropsychiatric. And one of the work my lab was involved in was to try to assess the impact of postnatal restoration in dystrophic animals. And so we first, in order to really look at the levels that would be necessary, we moved to local delivery. So we started doing intracerebroventricular injection, and not, because when you're doing systemic delivery with the tricyclo, there's only a tiny little bit that goes there and you need to treat for a long time. So it was better to do local delivery. So again, we started working with the MDX mouse model. That was even a little bit before bind. So injecting the oligonucleotides into the ventricles. And so we could see that this was a dose dependent. So we injected between 100 and 400 microgram of the oligonucleotides. And we could see a dose dependent increase of the exon skipping in the different structure. The oligonucleotides distributes quite homogeneously in the brain. This is one of the advantages of these molecules. Here it's a base scope analysis where you can really look at the specific skipped junction. So in red, the red dots here represents the skipped mRNA, where the exon 23 has been precisely excised, whereas the blue dots correspond to the initial mRNA. So you can really look where the oligonucleotide has worked and has induced that particular exon skipping. And this was associated to a dose-dependent restoration of dystrophin. So in that case, in the MDX, we were able to restore up to 25% of dystrophin in the hippocampus. And it was associated with a dose-dependent improvement in that fear response. So really confirming once more, if it had to be, that this particular phenotype was really due to central deficits of dystrophin. We then moved to another model because we wanted to investigate a model that would be more, that would look more like the patient and that would lack multiple isoforms of dystrophin. So we worked with the MDX52 model that lacks both the full length and the DP140 in the brain, also lacks DP260. And with work with Cyril and our PhD student Amel had revealed that this mouse model showed enhanced anxiety and severe impairment in fear learning. From a molecular point of view, this is interesting for exon skipping, so it lacks these two isoforms. The exon 52 is not present, and in order to restore the reading frame, you can either do exon 51 skipping, and in that case you would link 50 to 53, and that would rescue, theoretically, DP427. That would not rescue DP140 because the start codon of DP140 is coded in exon 51. So if you exclude exon 51, you cannot restore DP140. But you can also restore the reading frame by skipping exon 53. In that case, you would join 51 and 54. And in that case, in theory at least, you would be able to restore both isoforms, DP427 and DP140. We started with 51 because we had the compound ready and it was already approved in trials, and we wanted to see what eventually it could do to patients one day. So in the consortium, we had different chemistries of oligonucleotides available. So we had the tricyclo in our lab, but we also had the PMO that are approved in clinical trials. We had the methoxyethyl, which is the chemistry of nusinersen. So it's a chemistry also approved in the clinic. So we eventually wanted to see. So we did ICV delivery. Here's the direct comparison of tricyclo and PMO. Again, you can see both oligo distributes quite homogeneously in the different structures. Tricyclo tended to give higher exon skipping levels, so there were more oligo reaching the nucleus. And at that stage, I mean, we wanted to go as high as possible in terms of exon skipping. We wanted to be able to have as much protein as possible. So we tried to inject higher amounts of oligonucleotides, and for that, we tried repeated intracerebroventricular injections through the implantation of cannula, or we also tried infusion. So you can implant an osmotic pump that will deliver the oligo over 1 or 2 weeks, like super slowly. But in the end, none of the repeated administration really gave significantly higher amounts, or even the mini pump sometimes could, in some mice, they were really high exon skipping, but it was extremely variable. So it was not a very appropriate method. So we decided to go with the bolus ICV. So we published all these comparison of delivery routes, because we also tried interna cisterna magna. But that was also going more to the cerebellum than the other structures. We also compared with AAV-mediated delivery in adults, and I don't have time here to show it, but it was also giving a lot of variability. So the distribution is not quite as homogeneous as the oligonucleotides. So once we had selected the method of injection and the compound, so we went for bolus ICV injection of the tricyclo. We wanted to check when should we do the behavioral test. So we wanted to define the window of the therapeutic window. So we look at the biodistribution at 3 weeks, 7 weeks, or 11 weeks after treatment. And you can see that the level of oligonucleotides in the different structure clearly decreases with time. So the oligo is eliminated with time. But when we look at the efficacy of the exon skipping, it's actually quite stable. And this is interesting because it really suggests that the oligonucleotides that is eliminated is the unproductive fraction. So that's the part of the oligonucleotides that never reach the nuclei. So there are lots of oligonucleotides that get trapped in the endosome, or even that doesn't even get into the cell, that you would quantify here because it's in the tissue, but it's actually not doing this job. So it doesn't matter if it gets eliminated. So the efficacy looked stable, at least and up to 11 weeks. Regarding DP427 restoration, interestingly, we were not able to detect it at 3 weeks. So it really takes time to build up the dystrophin restoration. And it's interesting because in the muscle, for example, 3 weeks after a treatment, we normally detect it. So in brain, it takes a bit longer to accumulate. But at least between 7 and 11 weeks, the levels were stable. And this was also true by immunohistochemistry. So here, this is in the cerebellum, and here in the CA1 of the hippocampus. We can even quantify the levels. And locally, we can even, when we quantify in the CA1, go up to about 30% of restoration very locally. So that defined our therapeutic window for behavioral assessment. We knew that we could do our behavioral assessment between 7 and 11 weeks after the treatment. So that's what we did. At least at that time, we selected some behavioral assessment that were first compatible with each other in a small amount of time, that Cyril had validated, that were giving the strongest outcome. So of course we did the unconditioned fear test. That was the one of the main that we had previously used. And here again, we had an improvement with these levels of dystrophin. So yeah, I didn't mention we had between 5 and 15% of dystrophin restoration across the different structure. So this was enough to improve this phenotype. We also looked at anxiety using either the elevated plus maze or the light-dark choice, because MDX mice tend to show higher anxiety, so they do not, well, at least they go less into the open arms and they go less into the lit compartments. And there was an improvement in both cases with the treatment, so with the restoration of dystrophin. One of the last tests that we did was the conditioned fear memory, where we evaluate, and again, I'm going quickly because I know that Cyril is going to go through this test in a bit more detail than me, but we're trying to see if the mouse can associate a sound to an electric shock. And here, in the learning phase, we could see that in red, the treated mice were very close to wild type, so it was a full rescue of the learning phase. In the retention, where you look at the memory, there was only an improvement, where you see the red curves are really in between the control and the wild type. So this is an improvement. So we could conclude here that the partial restoration of the DP427 improves anxiety, fear learning, and fear memory in these mice, which was really promising for eventually future therapies in patients, suggesting that this may be reversible postnatally. Of course, we wondered, why is it only partial? you can always want more. So was it because the levels of DP427 are still very low? It's still between 5 and 15%. Is it because the DP140 is still lacking? As I mentioned, we wanted to explore also the restoration of DP140. And so we tried very hard to work on exon 53 skipping to induce the restoration of both isoforms. Unfortunately, and this is what we managed to still publish, even though it's negative results, it turned out to be very challenging in this particular mouse model. So the mouse exon 53 was very difficult to skip, and we had to use multiple oligonucleotides to target this, to manage to skip this exon. So we had to use 3 oligonucleotides to really skip it efficiently. And even though, so here, even with using 3 oligonucleotides, whether it be with tricyclo or PMO, we managed to get, it was quite variable. We managed to get some exon skipping in the hippocampus, but we never managed to get protein. And we still don't know why, but there was always less than 1% of protein restoration in this mouse model. Even though human exon 53 is much easier to skip, and it's in it's in the clinic, it's an approved drug. So it's the regulation of the splicing of this particular exon in mouse and human, which is different. So we had to use an alternative to restore DP140. So DP140 just about fits into an AAV. So AAV this is one of the limitations, they have a small packaging capacity. So you can only pack 4.5 kilobases. So with the promoter, DP140 is just about that size. So we did intracerebral ventricular injection of an AAV encoding the DP140 in these mice. When we do this, so this is the type of similar type of gene therapy approach that Alvaro showed yesterday with the DP71. In this case, with the level of AAV that we injected, we ended up with a very high upregulation, very high expression of DP140 in hippocampus and cortex, and much less in other structure like the cerebellum or the spinal cord. So this is why I meant by the AAV diffusion in the different brain structure is not as homogeneous as the oligonucleotide distribution is. Nevertheless, we assessed the anxiety in these mice. So we assessed the unconditioned fear response, and there was no effect. But that's we knew that the unconditioned fear response is not really related to DP140. We had DP140 was mainly related to anxiety, so this is why we looked at anxiety in these mice. And you can see that there was a tendency for improvement, but it's not statistically different from the control group that were treated with an AAV-GFP. But yet, it's not longer statistically different from the wild type, so there's a slight effect of this therapy. One final thing I want to show you is that we try to investigate earlier postnatal treatment, because of course we can also wonder if we normally treating the mice at young adults, like around 7-8 weeks of age. But what happens if we treat neonates? Maybe we would be able to restore restore better the phenotypes. So we tried to, we tried lots of different methods to restore dystrophin in the brain in neonates. One of the best is to do ICV injection. So you can do ICV injection in neonates of P0 or P1. So we did that with the tricyclo, and the first thing that we did was trying to look at exon skipping and dystrophin restoration at different time points. So when we looked at exon skipping, we had actually really high levels. So in the various structure, we went up to even 70% of exon skipping, and that was leading to nicely good 20% of restoration. But with the oligo, I mean, after 14 weeks, it starts to decrease, and that's typical for oligonucleotides. You need to re-inject. In this case, it means that if you need to re-inject ICV in the mice model, it means that you need to make the mice go through surgery again, and then for behavioral studies, it's not ideal. So that's when we wanted to use the AAV again, because we were like, okay, AAV is stable, so it should last. And in neonates, maybe the biodistribution would be a little bit better. So we again use that AAVU7 system. We did an AAVU7 targeting the exon 51. Again, doing ICV in neonates and the different time points. And in that case, we also had a very high level, going about 60%. And that was leading to nice restoration of dystrophin, but this was not go down. So it was really stable. So that's very encouraging. We had really high and long-term restoration of dystrophin. So we are currently with Cyril analyzing all the behavior, doing large groups of these mice to look at the impact that very early postnatal restoration of the dystrophin can have. So to summarize and conclude, I've shown you today that we can do intracerebral ventricular injection of oligonucleotides, and it induces widespread exon skipping in the central nervous system of these dystrophic mouse models. That partial restoration of the full length in the range of 3 to 15% improves anxiety, fear learning, and fear memory. And we believe that this is important for eventually future treatment, because it means that at least some deficits are reversible. We still have a lot ongoing, trying to investigate the additional restoration of other isoforms like the DP140, the early postnatal restoration. But so these are very important findings, because among the new generation of compounds that are being developed at the moment, not only the tricyclo, but there are lots of antibody conjugated compounds, and some of them may be able to also cross the blood-brain barrier. And I'm thinking about there are oligonucleotides that are conjugated to the transferrin to antibody targeting the transferrin receptor, and some have shown that they can cross and target also the brain. So it is important to do these studies and to trying to identify which deficits can eventually be postnatally rescued. So I want to finish by thanking my lab here in Versailles. And this is Ophélie here that has been working in Alvaro's group, and then Cyril's group, and then my group. And this is the entire BIND consortium, and in particular our collaborator, well, of course, Cyril, and also Francesco at UCL, and then LUMC, and all the sponsors, and you for your attention. Thank you. Yes, question?
Are there questions? Thank you so much. It was really nice. You know, you need to come to the microphone, because yesterday, unfortunately, the online people, they didn't hear the questions. So.
Hi, Aurélie. Thank you so much. Wonderful talk. Thank you for being here and for lecturing us about all this amazing stuff that you're working on. Can you tell us a little bit about the collaterals of this, the injections, if there are some, like problems with the therapy in general?
Okay, but you mean for the muscle, like the systemic deliveries? What is in the clinic, for example, or? Well, it depends on the generation. That's why the PMO, I think, are the only one to be approved. It's because they have no side effects. But they also are not very efficient. But because they are neutral, they're not positively charged like other oligonucleotides. They really do not have side effects. Among the new generations of oligonucleotides, I don't know if you've heard, but so Sarepta Therapeutics, which is commercializing the PMO, had a program on peptide conjugated PMO, and they were inducing higher levels of exon skipping, but they've just stopped their program because in the end they have issues in the kidney, for example, and they have elevation. Oh no, they have hypomagnesemia in patients, and initially they thought that it would be manageable, but in the end, it's not very manageable. So peptides, they tend to have complication. Antibody, so far in the results that have been released in press release show a nice safety profile. They don't show any adverse events, but we need to see further. I guess, yeah, the trials will tell us. Generally, people are being careful with kidney because these molecules, when they are injected intravenously, they do accumulate a lot in liver and kidney. So this is where people are being careful. Yes. In mice, yeah, in mice you can check for biomarkers. So the type of biomarkers that we had looked for, urinary biomarkers. You look at KIM1 or renin, and if they start to go up, you know that it's before the lesions appear. Then the biomarkers tend to appear. You can look at careful histopathology and of course, anyway, before any of these drugs go into the clinic, they go through regulatory tox studies. So they get injected much higher dose. So.
Just curiosity. Thank you for the wonderful talk. It's a very rich area, and which I'm not so knowledgeable about. But I was curious about the behavioral tasks that are being tested. Why only aversive stimuli are being dealt with? Why not positive reinforcement tasks in learning and things like that?
This is not a question for me, Dora. This is a question for Cyril. I'm only applying the test that he tells me to are working. He's the one characterizing all these phenotyping. So Cyril.
Yeah, but it's not that we have not tried other tasks. We tried with positively reinforced tasks. There was some phenotype, but they were not so easy to visualize. The most important phenotypes in those mice are associated with aversive stimuli. That's where we have the biggest difference with wild type, which is for us, for this type of approach, with a therapeutic approach, it's better to have a large difference to start with. Then you can see if you have even a partial improvement is more easy to see. You can see some deficits with other tasks, but they are more subtle. And also, most of the time, there are learning tasks that require, like, for example, 2 weeks of training, which is not always ideal for this type of therapeutic approach, where the time window is around 7 weeks after treatment, and then the treatment effect drops down. So we chose, for those several reasons, we have chosen the alterations that are associated with aversive stimuli. It's obvious that in these mice, one of the main phenotypes is associated with their reactivity to stress, anxiety, and all the, I would say, emotional learning that goes with this kind of aversive stimuli. So the bigger phenotypes, more easy to see an improvement, and also shorter experiments to be able to do several experiments during the therapeutic window, which is sometimes short, like 7-8 weeks after treatment only. So that's the main reason. But there is some, yeah. For example, we found some memory deficit in tasks. For example, in the water maze, the mice, they learn well, but then they have a slight deficit in memory when we ask them to recall the learning. But then you need already 1 week of work to see the deficit, and the deficit is small. Also, if they learn, for example, to get food by food reward by pressing a lever, they have a small delay in learning. But the delay in learning is much more important in fear conditioning, for example. So with aversive stimuli, it's always bigger. So that's...
does not mean that the deficits they have are restricted to those functions, right?
No, there are others. There are even social behavior deficits, but this I don't know. It's not obvious from the observation just in the animal facility. We put cotton sometimes, and they make a nest. So, but social interactions are altered. So, I mean, it's in our plan in the future also to diversify a little bit more the task. But it's always more difficult when you have subtle differences to see if there's only a partial improvement with the treatment. So. But yeah, we will try other tests in our future project.
And I didn't have time to mention, but one of the reasons that I'm here and that we are working together is that because the tricyclo crosses a little bit the blood-brain barrier, they also cross slightly the blood-retinal barrier. And then we have seen a little bit of exon skipping in the retina after systemic treatment. And this was the reason why we initiated, or part of the reason, that we thought, okay, eventually a treatment one day, like the tricyclo in patients, if we can show that the level of crossing the blood-brain barrier correlates with the level of crossing the blood-retinal barrier, then eventually ERG could be used as an outcome measure to know how much has reached the brain and also how much has reached the muscle without having to do a biopsy. Of course, that relies that maybe that's only applicable for a compound that will target both or like cross both barriers in a similar manner. So, for example, the compounds that I mentioned are antibody conjugated to the targeting the transferrin receptor. They might have the possibility, the ability to cross the blood-brain barrier, but not the blood-retinal barrier. So you would not use ERG to look at brain uptake for these particular compounds. So this would not be something used for all the compounds, but eventually for this type of compound, this could be used. So since then, we're trying to look at how much dystrophin we can restore in the retina, and with that improve ERGs. Yes. But yeah, we're not there yet. Yes, of course.
Thanks for your lecture. I have a question. When I saw in your one behavior experiments in neonatal animals, I saw that you tested until 40 days, 14 days. Yeah, 14 weeks. Oh, do you have plans to have a study that have a longer time to do if the effects of the gene therapy continues?
Yeah. Sorry. Thank you. Yeah, we can look at longer time points. This was already 14 weeks. Yes, so this was at least covering the period of behavioral testing that we would want to do. Of course, we can look further, but we're quite confident that if the levels are stable with the AV at 14 weeks, they would still be stable. I was actually more scared of losing some because of the growth. So AV is stable, but of course it gets diluted when the cells are growing and dividing. So injecting at P0, we could have lost some AV along the way, but it was quite positive to see it's very stable.
So these treatments approved, they don't cross, right? And do you think the clinicians are looking for improvement in cognition? I mean, do they expect that the systemic improvement of heart, muscle would also help the central nervous system? And can you see in mice, in animals, if simply improvement, cardiological improvement or muscle improvement could help the brain?
Thank you. It's a very good question. So the first part about the currently approved treatment do not cross, and there's no outcome measure anyway to look at behavioral improvement or cognitive improvement in patients. This is also one of the objective, I think, one of the research line that people are trying to do is having proper outcome measures for eventually future trials that will, improve cognitive function, how to really measure it in patients. And the second one, can eventually muscular or cardiac improvement also have effect? That's a really good question. We actually wondered at some point if there was not a contribution, at least, of the peripheral improvement in the behavior that we were seeing. And what I didn't have time to show is we wondered if the partial restoration that we could see could be further improved with peripheral restoration. So we combine with a peripheral treatment where we also restore dystrophin in the brain and the cardiac muscle. And we didn't see further improvements at least in the test that we did. I don't think in this series of mice we tested all the behavioral tests possible, but in the one that we typically do, we didn't see further improvement. It was it's interesting because, for example, if we take the and there I'm going to point at really, questions that we have, but in the fear response test, the response that we had with the tricyclo treatment systemically administered, when I show you the initial work, where with the high dose at least, the fear response was fully corrected. So the levels of the freezing response was down to wild type levels, even though we know that we had only 2% of dystrophin restoration. And when we do it by ICV, where we had 5 to 15%, we had an improvement, but it was far from being to the wild type levels. So this is the type of question where we wondered if there would not be also a peripheral contribution. That we're still wondering. The experiments that we did prove the opposite. But there are other works that suggest that there can be a contribution. Ideally, you want to restore both. So we would not want to restore only the CNS in these patients, so they would also have muscle and cardiac restoration. So hopefully, if there is a peripheral contribution, then we would see it. For the CNS or for muscle? For CNS, it looks like the deficits are not progressive. For muscle, of course it is. For muscle, clearly the earlier the better. Although, and I think this is a point of discussion with AAV gene therapy, I didn't mention all the microdystrophin work here, but there are also AAV microdystrophin treatment approved by the FDA. And there's the question of the dilution here again. So you want to treat the patient as early as possible, but because you cannot re-inject, if you inject eventually even a baby because somehow it has been diagnosed, eventually because there's a prenatal diagnosis or anything, you have a chance of maybe diluting the AAV with age because the boys are growing. So it is a tough question, I think, you want to treat as early as possible, but you don't want the AAV gene therapy to dilute too much. And for CNS, maybe there's... So far, from what we've seen that we could rescue some deficits even in adult age. But, we cannot exclude that there are things that are developmental. So maybe even postnatal is not enough. But we have nothing to to evidence this so far. That's ongoing work. Oh, Cyril.
Questions, but maybe it's difficult questions, but you will tell us. You show in your talk that there was a lot of work to finally achieve some efficient molecules. You change the chemistry, you change the delivery route, so it's a long process to find the best way to improve the thing. But you also mentioned some experiments showing that there are some problems sometimes that are not so easy to understand. Maybe at least I do not fully understand. For example, why a mutation induce an imbalance in the 5' 3' production of RNA? And is this general, or is there some specific mutations that are more specifically doing this? Like, is it the same if you have a point mutation or a deletion, or on the position of the mutation? I mean, is it a general problem? It's maybe just me that didn't understand, but maybe you can, with simple...
It's a very good question too. Well, we haven't looked at so many different mutation profile. I know that it's been published that it's not necessarily due to deletion. So I think deletion have been compared to point mutation, and it was seen in both cases. What we have seen is that there's an impact on the position of the mutation. So the more distal, the worse it is. So in the MDX52, than in the MDX23, there's even less RNA available. So the imbalance is even stronger. So the curve is it's even worse. So there's very little dystrophin, little pre-mRNA at the 3' available. And this is also why we have this one of the hypotheses why we have this discrepancy between the exon skipping level and the protein restoration. And so we think it is a very important point to raise in the DMD community and in the therapeutic community, at least exon skipping, because the gene therapy don't have that problem because they do not target the RNA. But in trials, it's often seen that there's very little dystrophin restoration, even though sometimes they detect significant amount of exon skipping. But to make it even like as an image, if you skip 100% of your mRNA, you would think, wow, this is great. But if you've got only 5 copies of mRNA, okay, there are 5 skipped copies, but it's not 5 copies of RNA that's going to give you a lot of dystrophin protein. You need thousands of copies. So that's how exon skipping percentage sometimes is not really an appropriate measure for efficacy. So it's really the protein that's important. And this imbalance is not a well-recognized problem yet in the community. It's not fully understood. There's this work from the LUMC group and Pietro Spitali that have characterized epigenetic marks on the DMD gene, and that clearly influence the transcription rate. So that's a possibility. And that's something that we want to further investigate.
Is it seen in human patients?
Yes, well, yes, in biopsies and in human cell lines, and from different deletions, different mutations.
So means that the more severe mutations that are distal are going to be the most complicated cases to cure. Well, that's an important point to know. Another question, same type of question maybe. Why you need to use multiple RNs to skip exon 53 and not when it's for skipping exon 51? So why is it better to have several different AONs instead of increasing the concentration of one, for example? And why does this happen in mice but not in humans?
Well, we haven't completely identified this. The exon 53 in mouse is differently regulated. The splicing of exon 53 in mice is differently regulated. So the splicing signals are a bit different, and it has weaker signals. So it is, let's say, a bit weakly, less well defined. So you need to block several signals in order to skip it. Whereas the human exon 53 is a stronger signal. So if you block one, it gets eliminated a bit more easily. Well, that's a metaphor a little bit, but that's how it's regulated. And this is why you need to target several. But this would not be therapeutically relevant. We only did this because we wanted to achieve exon 53 skipping in order to restore the protein, but it didn't work anyway.
Maybe talk a little bit about the use of humanized mice.
Oh yes. So we as an alternative. So there's a humanized DMD mice that exist. So the Netherlands has produced it. The HDMD so carries the entire human DMD gene, and they've also made deletion of exon 52. So there's a humanized deletion 52 mouse that exists. Currently, it's crossed with an MDX. So it means that in the brain, it still expresses some mouse isoforms. So for example, for us, when we want to do, because, okay, go back there one step. If you want to do exon 53 skipping, that would work. We can skip a human exon 53. So for us, it would be easier to work with the human sequences. We know that, well, in theory, it should work. So we want to use this model, but if we inject the human exon 53 sequences in the brain in this mouse, it still expresses the mouse DP140. So we need to generate a new model and cross that model with the DMD null that doesn't express any mouse isoforms to perform all these behavioral tasks. So this is something that's also ongoing. More work for us to do. Well, I hope so. Yes, yeah. Normally the human exon 53 skips well. Thank you.
We can talk during the coffee break, okay? Okay, thank you very much. So for the people online, we say goodbye and we meet again tomorrow. Thank you.
É o que eu tô pensando aqui. Essa é a pergunta. Verdade, a gente cometeu